Basic components of cell theory timeline starting from Robert Hooke. Proposed in 1838, over 150 years after Robert Hooke's Micrographia. Cell theory is the foundation of modern biological sciences. As microscopes became more sensitive and observational techniques allowed for the viewing of internal cellular structure, the theory expanded, but the original three tenets have remained the same. Cellular theory defined classical cell theory, first proposed by Matthias Schleiden and Theodor Schwann, consisted of three primary points. 1. All living things are made up of cells. 2. Cells are the basic units of structure, function, and physiology in living things. 3. Living cells can only come from other pre-existing cells. 4. Modern cell theory adds 2 additional points. 5. Cells contain and pass on hereditary information during cell division. 6. All cells are relatively the same in relation to chemical composition and metabolic activity. The significance of this seemingly basic concept cannot be denied. At the foundation of almost all sciences, including biological sciences, chemistry, biology, and medicine, is cell theory. This turning point proved on an elemental level cells are similar in the way they function and reproduce, and with the advent of more advanced observation techniques, the differences between cells relates to genetic makeup. Robert Hooke, describing the appearance of a thin layer of cork tree, one of the many microscopy documentations contained in the 1665 publication Micrographia. Robert Hooke is credited with the term cells. Hooke likened the magnification of the cork's box-like pores to a monk's living quarters, known as a cell, of Latin origin. Celia is translated as storeroom or small container. Hooke did not identify any internal cellular structure, only the external walls of the dead cork cells. Along with other scientists of that era, he thought the empty spaces might transport fluids in plant life. Contributions to the field of physics include Hooke's law, where he explained the physical properties of linear elasticity as it related to a spring. He also made significant advances to the design of astronomical instruments such as Gregorian reflecting telescopes. Hooke died long before the development of cell theory. Cell structures under the microscope. The microscope was improved and modified for better observation of different cells and microscopic organisms. As a result, cell theory was created and modified to be what we know today. The physiological and biological study of cells focuses on cellular structure and function. Cells function individually, yet also come together to form organs and play a role in respiration, elimination, and digestion. At the foundation is cell theory. All living organisms are made up of cells that can only come from pre-existing like cells. Therefore, to understand behavior, genetics, or disease, one must study cells. The two primary classifications of cells are prokaryotic, which contain domains of eubacteria and archaea, and eukaryotic, which encompasses protists, plants, and animals. Prokaryotes, literally translated as before the nucleus, contain the domains of bacteria and archaea. Unique to prokaryotes are flagellum and pilus or fimbriae. Some prokaryotes contain organelles through enfoldings such as cilia, centrioles, and ribosomes. The internal and surface structure of a bacterial cells include nucleoli, ribosomes, storage granules, endospore, capsule, outer membrane, plasma membrane, cell wall, periplasmic space. Prokaryotes replicate through binary fission. Eukaryotes are more complex organisms and, with a few exceptions, contain membrane-bound organelles. Common to plants and animal cells are cell membranes, cytoplasm, and a nucleus. Know that you're coming back home.